Very good morning to you. We are going to start the important topic. Actually, we already started the this particular title translation, and I also given a significance of a genetic code as far as procedure of translation is concerned. So, in summary, once again, I am going to repeat about the genetic code, and then we are going to start the actually what is the translation is. Various features were already discussed in our previous lecture. Once again, just a point of a summary or point of revision, I am going to explain one by one. First thing is that the codon genetic code consists of the nucleotide triplet, three nucleotides on the mRNA that is coding for the one amino acid. This is the concept which is generated by the efforts of so many scientists like Argovind Khurana. The next point is that the genetic code is non-overlapping. What it means that each nucleotide in mRNA belongs to just one codon, except rare cases where the genes overlap and nucleotide sequence is read to different reading frame. Means it is not there is should be not overlapping as far as two sentences are concerned. For example, in case of a book, we say that we refer that there is one sentence, full stop, then another sentence. We never read that the some part from the first sentence and another part from the another sentence. We never read that. Similarly, without there are several exceptions are there as far as genetic code overlapping is concerned, but the majority times genetic code is a non-overlapping. Then next is the genetic code is a comma free. What is the rule that always their triplet is considered? Suppose if one is get removed, then it is not so that only two will get read. Immediately third will be there. Means there is nothing like a comma like this. Means one word is removed. For example, in the same example I am taking that in sentence. Suppose if I vanish one letter, so we generally read the another word as it is. Okay, so for example, if I am saying that the microbiology is important subject, in M is removed out. If M is removed out, then there is a still we read as the microbiology we read and M is not there, but the important subject. But in case of genetic code reading, M is not there, but M is get at microbiology is attached to the another word, and in that way, overall total sentence is get same. Hence, for genetic code is a comma free. Then the genetic code is generated. Much more elaborate this particular point that the amino acids are specified by the more than one codon. That also we are considered by taking the visualizing the chart. The next is that the genetic code is ordered. I explained this particular thing as the genetic codes are a non-random. Why it is so referred? Because multiple codons for the given amino acid and codons for the amino acids are similar chemical properties closely related to this. Okay, means whenever the you compare, there is a particular relation between a codons and chemical properties of a amino acid. Therefore, this has a particular evolutionary significance. That point also clear to you. The genetic code contains start and stop codon. AUG is the initiation codon, and there are also stop codon. We have precisely deal with this particular concept in coming lecture. What are the stop codon? How they are responsible for the terminating the translation. And the genetic code is nearly universal. Actually, in so many books, it is mentioned that the genetic code is universal. But we also know that there are several the mitochondrial systems are concerned, particularly humans or the mammalian. They doesn't obey the genetic code rule. Hence, we have used the word nearly universal. It's not applicable to all. So that's all regarding the genetic code. And now it is possible to discuss regarding to the process. We already know that this is the third process. Already we are aware that the from this particular DNA, mRNAs are synthesized from along with mRNA or RNA precursor tRNAs are there. They orient themselves and they form the three-dimensional structure. Three-dimensional structure. Okay, and then all these assemblies are combined with each other, and then they are contributing for the synthesis of a protein. In that, ribosomes are very much important bodies, and mRNAs are also important, and tRNAs are important. mRNA's role is direct because it gives the instruction what should be the structure of a protein. Okay, but prior to that, this particular translation, there is the activation of a particular amino acid. That amino acid tRNA should be attached, and then it can enter into the procedure. Okay, that is the this particular thing is the translation. Actually, in case of bacteria, there are several proteins which are also synthesized in absence of ribosome. Okay, ribosomes are not required. Generally, 10 to 12 amino acids are there. These are connected to each other by means of enzymatic process. So that is not a translation. Means without ribosome also very rarely, particularly it is detected in case of bacteria, that is also possible. I suggest that you should discover such a proteins, short peptides, which are synthesized in absence of ribosome. It is a home assignment to you, to all of you. Search out the such a peptides, short proteins, which are synthesized enzymatically. Enzymatically means without taking help of a ribosome. Here, one important difference I already given that the in case of a prokaryotic, the gene whatever transcript is synthesized, that transcript is directly available for the synthesis of a protein. Means transcript which is generated from the transcription, it is available for the translation. And one codon gives the amino acid one, the second number codon gives the amino acid two. In that way, it is a continuous one. But while in case of eukaryotic system, what happens? There are a gene is not a continuous one, just like a prokaryotic system. There are a exon, intron. Then intron are removed. We already clear this concept that due to the RNA itself, the cell splicing is there, and intron are get removed. And in that way, the all these codons, then these are connected because this unnecessary part is removed out. Okay, wherever the unnecessary part is there, is removed out, and then this particular processed transcript, okay, this is the primary transcript and this processed transcript, then this processed transcript is available for the translation. So this is the very important difference as far as the prokaryotic and eukaryotic system is concerned. In prokaryote, such kind of exon intron interventions are not. That is a very important point. Now, keep in mind, now in case of prokaryotic systems, this transcript is ready, and in case of a eukaryotic system, this process transcript is ready. We are focus particular discussion as far as the prokaryotic system is concerned. Whenever time permit, I will elaborate what happens in case of eukaryotic. The first step as far as this translation is concerned, actually it is synthesizing the activating the amino acid which is required, because for the protein synthesis amino acids are required. Proteins are nothing but the polymer of amino acids. There are 20 amino acids, and for the 20 amino acid codons are also there. But first of all, first thing is that these amino acid, okay, means it is applicable to all, R is different. Whether you see the PowerPoint presentation, there is no problem. Okay. Now, in the first step, what happens? There, this particular thing is referred as the activation of amino acid. It is the two-step process. Activation of amino acid is the two-step process. In the first step, whatever amino acid is there, it is get activated by means of utilizing the energy from the ATP. Means it is a ATP consuming process, and this is a in this step, amino acid react with ATP and forms the amino acid AMP. Here it is shown in diagram. ATP phosphate group is released, and now this resultant structure is get formed. So this is nothing but the this is the amino acid here. It is get attached to this ATP's moiety, but the name of this particular molecule is, as the phosphate group is removed, it is referred as the amino acid adenylate. So this amino acid, acyl adenylate, this is the adenylate part, and this is the amino acid. So this is the, now one thing keep in mind, there is the, this is the enzymatic reaction. For that purpose, the enzyme which is required, it is referred as amino acyl tRNA synthetase. And these amino acyl tRNA synthetase are the highly specific for the amino acid and tRNA. How the tRNA structure is the same, but how the tRNAs are different? when I'm saying that there are a different types of tRNAs are present, but generally all the tRNA has the same configuration, three-dimensional structure is the same. But still, I'm saying that there are different types of tRNA there. What it means?
Active site different assignment.
Which, which side?
Active site. Active site.
It is not enzyme, Krishna. It is a tRNA. I'm asking about the tRNA. tRNA, how the tRNA will be different in the in the cytosolic. tRNAs are different with respect to their anticodon site.
Anti-codon.
Okay, the triplet of anticodon which is present at anticodon site, it is a different. Okay, and in that way the tRNAs, different types of tRNAs are there. Okay, now what happens? These amino acid tRNA synthesis are specific, means they for the pairing with amino acid with a particular tRNA. Okay, because we already know there is a correlation between the anticodon and a amino acid. So this particular conjugation is done by the amino acid tRNA synthesis. Means different tRNA amino acid RNA synthesis are required for the different amino acid and tRNA. Okay, here how this particular amino acid and particular tRNAs get coupled, because this is the specifically encoded by the process by the specific enzyme, that is the amino acid tRNA synthesis. Then what happens in the second step? This particular complex is still there. Actually, enzyme is there, amino acid synthesis is there, and these moieties remain attached. Okay, first it is get activated by means of ATP, but still everything is available on the that enzyme. And in second step, the amino acid, okay, AMP intermediate, this is the amino acid AMP intermediate, it is not released, it is remains attached to the enzyme amino acid tRNA synthesis, and in second step the amino acid group is transferred to the 3' end of respective RNA. At the tRNA, you know that this is the amino acid site at the 3' end. This amino acid, which is part of this particular amino acid RNA system, that is shifted there. This particular 3' it is get bound to the amino acid, and in that this particular depends on the anticodon. Anticodon is located here, and depending on the amino acid is there. But who will going to decide that depending on this anticodon, this particular amino acid will be bound to this tRNA? This is done by the enzyme amino acid tRNA synthesis. Means for the different tRNA amino acid, different enzymes are there. That you should keep in mind. Now here so many tRNAs are ready. tRNA with the amino acid. This is referred as the activated tRNA or charged tRNA. Why? Because now amino acid is get coupled with them, and now these are present in the cytoplasm abundantly. And here, just like transcription and replication, translation process also follows the three steps. First is the initiation of translation, another is the elongation of translation, and last is the termination of translation. In the initiation, what happens? There should be first assembly of a mRNA and a small unit of a ribosome. Okay, so there should be the assemble, means first in the initial stages these are the bit couple. But how these particular two things are get assembled? Don't expect that the ribosomes are always ready. Whenever the protein synthesis is required, then and then aggregate ribosome molecule is get from small unit and large unit. Otherwise, everything is available in a diffuse state. Diffuse state means the small unit is diffused, large unit is diffused, mRNAs are diffused. But just for example, if mRNA is now ready by means of transcription, and they approaches to the first the small subunit of ribosome. Okay, and we know that this particular transcript is always read from the first codon, that is AU. But how this particular assemblies takes place? This is due to the one important sequence that is referred as the Shine-Dalgarno sequence. So this is the present on a mRNA. This is the present on the mRNA, and now a complementary sequence is available on a 16S rRNA. 16S rRNA is present on the small subunit. Okay, so there is a complementary structure. Now keep in mind this SD sequence or Shinez Algorithm sequence is a different for the different organism. Means in the different organism the sequence might be different. Here you will also find different concept. But many times it is a sequence is a 5 prime to UAAGGAGG. But modifications are also there. But many time, majority of time, this is the sequence. Means this is the 5 prime. Here the UAGA, and this is always associated with the AUG. Here from here the translation is going to start, and just towards the 5 prime end, this SD sequence is there. Hence, for the mRNA, which is freely available, just recently synthesized by the transcription, now approaches to the smaller unit of the ribosome. Okay, and in a smaller unit, we know that there are several different types of RNAs are there, particularly 16S RNA are there. So they form the complementary structure, and in that way, the structure is get formed. Mean initially mRNA and small unit is get. Then another activated tRNA. How that tRNAs get activated, that already we are known. This is the two-step process, and so many RNAs are. But first, the tRNAs with code for the AUG code for the methionine. Hence, for activated tRNA is there. Now these tRNAs comes. Now here there is a complementary sequence. SD sequence is present on the mRNA, and anti-SD sequence is present on the ribosomal subunit. In that way, the both are get coupled now, folded to each other. Now the tRNA comes here, and you know that the anticodon specifically bound to the AUG. Okay, and hence for at this particular smaller unit, this complementary tRNAs get bound, which has the AUG as a codon. So this is the initial complex. Okay, I'm once again elaborating this particular initiation complex, how it get formed. But keep in mind there is systematic process. First mRNA small unit combines, then tRNA comes. Now, then in later on picture, what happens? A larger unit of ribosome is coming. Here also keep in mind the magnesium ions are also plays very much important role in order to assembling the ribosomal unit. So here there are three sites are there: E site, P sites, and A site. Okay, A means acceptor site, P means P site, and E is the exit site. Okay, these are not a typical labeled site. This is just for our convenience. We are given the name. Actually, at a time, as far as this result is concerned, only two tRNAs can bound. Okay, at any instance, this particular space can provide the binding of only tRNA. But for our convenience, we have labeled this particular part as the E site, P site, A site. A is for receptor, P is for peptide, and E for exit. Okay, but keep in mind that at a time three tRNAs are never bound. Only two tRNAs can bound. Okay, so what it means at acceptor side the amino acids are get bound, then at peptide side the peptide will synthesize, and there is the translocation movement of ribosome over the mRNA is there, because typically it going to read these things, and hence for this location is get a change. How it happens in case of elongation I will explain, but just keep in mind this is the this particular large unit is for the support, because it support the hold the tRNA, and this particular initiation assembly is prepared. Okay. Then question arises whether the SD sequence is present in case of eukaryotic system also. The answer is that it is not directly you can say that these are the SD sequence are present, but same sequence is present, and in case of eukaryotic that sequence is referred as the Kozak sequence. Okay. Kozak sequence is present in case of eukaryotic, means that have the different modified structure, while in case of bacterial system, in case of prokaryotic system, SD sequence is present. Means SD sequence is present on the mRNA of a prokaryote, and Kozak sequence is present on the eukaryotic system. So what happens as far as the initiation is concerned, there is an assembly of a 30S initiation complex. I already given this particular idea that the 30S is the smaller unit, and that forms it. And for that initiation, several proteinaceous factors are required. Okay, and these are referred as the IF1, IF2, IF3. Okay, so this is the abbreviation of initiation factor, initiation factor 2, initiation factor 3. Why these are labeled like that? Because they are required in the initial step, initiation step of a translation. Hence, these are referred as IF1, IF. So they are important for what is the significance? They are holding the mRNA, holding the 30S ribosome, proper orientation they mediate, and in that way, due to their particular efforts, the binding of 30S ribosome, because I already given that there is a particular sequence, SD sequence and anti-SD sequence, but the proper holding, proper orientation is also important. Then and then we know that the key and lock has the complementary structure, but key should be properly inserted. Then and then it will going to bound. For that purpose, we have to take a effort. Such efforts are taken by the initiation factor. The initiation factor 2 recognize form a methionine tRNA. Once they are get bound, then the recognition of methionine. This is particularly we can predict which is the first tRNA. Why? Because initially AUG is the codon, hence for it is always the formulated methionine tRNA. Okay, that is recognized by the IF2. IF3 is also involved in recognition of the start codon. So IF2 is recognizing the tRNA, IF3 is recognizing the start codon AUG and matching anticodon end at the initiator tRNA that are mediated by them. The IF3 prevents the 50S subunit from binding. When these are binding at that time, the 50S initially 50S is not in picture and it is not bound, and this is prevented by means of IF3. Okay. Then as the correct tRNA is available, once the proper tRNA is available, then and then 50 RNA will be come in picture. And this particular complex is referred as the 30S initiation complex. Henceforth, that can be assembled. Now what happens? The function of IF3 is finished now, and then IF3 is get removed, departed. Okay, and you know that the IF3 is preventing for to the 50S, and now IF3 get removed out from that assembly. Now it is the time to bound to the 50S to the initiation complex. Initiation complex means 30S plus mRNA. Okay, the initiation factor IF1 and IF2 are also released. Why? Because they are just required for the initial consequences. Now their role is not there. Now at the ending of the initiation step, a complete 70S initiation complex is get formed. 70S, why I am referring, because 30S is already bound, then later on 50S comes when the IF1, IF2 and IF3 are get removed, because IF1, IF2 they are properly orienting these all constituents, and even tRNA is get bound, and then they all got get removed, and that allows 50S to bound. Now in the picture 30S, 50S, we generally give this particular assembly as a 70S. Hence now at the last the this particular complex is referred as the 70S initiation complex. Okay, and the energy which is utilized for the proper orientation of these mRNA or 30S and all these things, it is in form of a GTP. Okay, it is not ATP, it is a GTP. In for the activation of the tRNA, ATP is required. In the initiation process, the GTP is required. Now, this is the elongation process. In the elongation process, what happens? Here, keep in mind, this is the structure. Here, the acceptor side means here the tRNA comes here, then it translocate. Okay, and once it get moved, at that time when the tRNA is here, at this particular thing, the peptide bond is synthesized. Here, the peptide bond is synthesized. Now, this is done by the peptidyl transferase enzyme. Actually, although it is an enzyme, but keep in mind it is a RNA. This is the exceptional example. I already given that the all enzymes are not a protein. There are several RNAs. These are referred as the ribozyme. So this is the good example of ribozyme, the peptidyl transferase. Actually, what is the peptidyl transferase? Peptidyl transferase is the 23S rRNA of a large subunit. Within a large subunit, a 23S RNA is there, and this is nothing but the peptidyl transferase. We are given the name just like an enzyme, but this is not a protein molecule, it is RNA. And these peptidyl transferase is response to synthesize, connect, or form the peptide bond. Okay. Here, already this particular tRNA is there, and now another tRNA is get bound. Depending the binding of this particular tRNA is depending on the codon mRNA reside which tRNA will be going. All the tRNAs are available now, and in that way, whenever their number comes, depending on this codon, they are going to bound. Once these two tRNA are there, from this amino acid is shifted to this, and they are connected by means of a peptide bond. And when it moves, system goes, then a free amino acid means, for example, here it goes here, and he whole small peptide is shifted on this, and this becomes a free. Once again it is available for the charging. This is now uncharged. Depending on the requirement, it is going to charge once again, and whenever it charge comes and waiting for its return. Means which tRNA should be specifically bound to here, that is decided by there, and for the binding purpose this particular ribosomes are there. Now in that way, this amino acid, when here it comes, all these small peptide is shifted here, and in that way now tryptophan come here. Now the phenylalanine come here. In that way it is continuously going, and these are connected by means of a covalent bond, peptide bonds, and these peptide bonds are formed by the peptide transferase, which is the RNA. So that's all regarding elongation. In elongation, same thing is happens. Once the now in this case there is no need of initiation factor, because initiation factors are get released. Their role is here more systematically it is shown. For example, here formyl methylene it is bound to the AUG. Okay, this is the happens in case of initiation. Now here GTP is required, and now another amino acid which is coupled to the tRNA it comes. Okay, depending on the sequence the tRNA will come now. And now here already one formyl methyl amino acid is there, and another second number amino acid is there. Now what happens when both are available? Now this is going to shift. There is the movement of a ribosomes over the mRNA is there, and hence what happens, due to this the location is get changed. Okay, so here it comes at the P, and this particular formyl methionine goes here. Okay, it also requires the ATP. Now, and meanwhile, what happens? The formyl methionine is shifted on this, and in that way, now this structure is there. Once again, same thing will be happen. This will get removed out, okay, and another will going to bound, and in that way, at a time, only two tRNA is there, but only the location is get changed. Okay, keenly see what is actually happening here. First, here tRNA is get bound, then it is the another tRNA. Why it is referred as acceptor? Because it accepts the new tRNA which constitute the charged tRNA, or tRNA with amino acid. And then there is a translocation movement, little movement of the ribosome that dislocate the position. And here this picture comes here. Now it get removed. Until and unless it not get removed, the another will not there, because I already given, for the sake of convenience I am showing this particular three sides. Actually there is not much more space is there. At a time only two tRNAs can bound. Once it get removed out, then another amino acid 3 containing tRNA comes. Then same thing is happening so many times, that is referred as a elongation. Okay. Here also elongation requires two factors, EFT and EFG. Here it is shown. In the first step EFT is required, and in another EFG is required. The EFT consists of the two proteins. Okay, these are the proteination factor, EFTU and EFTS. EFTs consist of the two monomers of the two peptides, EFTU and TS. The incoming activated tRNA is delivered to the ribosome and installed into the A site by the elongation factor, particularly EFTU. So this EFTU plays a role to install the newly coming tRNA at the A site. and this requires the energy as it is shown in diagram. It is utilized in form of GTP. While EFTS is responsible for exchanging the GDP left bound to the EFTU for the fresh GDP. Okay, proper coordination with the enzyme, and along with that enzyme, the GTPs are also coupled, and for that purpose, these proteinaceous things are. And the another elongation factors means here for the binding of the tRNA to the A site and involvement of GTP that are mediated by the two proteins EFTU and EFTS. Okay. Now another factor, that is EFG. This EFG is required for the translocation. Translocation means movement of ribosome. For that purpose, EFG is required. So this EF is elongation factor. In initial cases, IF name are given. That means the initiation factor. This is the EF. Okay. So in that way, this process continues, whatever shown in elongation. But what happens at particular point while reading, it comes at such a position where either UAA, UAG, or a UGA is present. UAG, UGA, or UAA. If such a codon comes, for that codon there is no tRNA available. Whenever such codon comes, at that time to binding to this particular anticodon containing such tRNA is not available. Means it gives the message to the system that the here you have to stop. Okay. For the stopping, this is the first signal that there should be the occurrence of the nonsense codon. Okay, I already given the name. This particular also recognized by the another alternative name, Zamber, Opal, Archer. Okay, so they should be appear in the system in mRNA. They should come there. Then and then there is a DOTM. But along with that, there is also requirement of RF factor. Okay, the RF1 has the ability to recognize UAA, means whenever the UAA comes here, the RF1 comes. Whenever the UAG is, then at that time RF1 comes, and UAA and UGA can recognize by the RF. These two factors contribute the dissociation. Whenever such a assembly comes, means whenever the mRNA nonsense codon comes, this particular respective RF factors are come there. Okay, and along with them RF3 factor is also there. Okay, and these RF3 whenever comes, means they are sequentially, first RF1 RF2 comes, and depending on that RF3 comes, and due to this what happens, RF1 and RF2 has get dissociated. So these RF3 is actually responsible for the dissociating the all structure. What is the message here? That there should be this protein synthesis should stop here, because whatever all amino acids are get connected to each other. Now there should be the stop, and hence due to these RF factors, what happens? There is the dissociation. Dissociation means every part is get detached from each other. Whatever assembly formally get formed, because protein synthesis is complete now, and in that way the RF3 and ribosome recycling factor. There are so many ribosomes recycling factor, because for the dissociation of ribosome recycling factors are required, and they are responsible for the detachment of these tRNA, and then the synthesized peptide. For that purpose, this RF3 is working, and for the dissociation of this 30S and 50S, ribosome recycling factors are required. Okay, and once again, the assembly becomes just like a initial phase. Initially, you know that there is a everything is separated, and whenever time permits, if the proteins are required for the synthesis, once again same thing is there. Actually it is quite difficult to you, but here it is electron microscope. These are the globular molecules are the ribosomes. Okay, so this particular photograph is taken from the silk worm, Bombyx mori. Okay, how the silk is synthesized, and in that way the arrow showing that the particular things here, initially the sequence is less, but later on you will find that the length is increased. Okay, so in that way this is the mRNA, and it is passive. Now what happens? So many ribosomes are there in SCO. At a time, so many copies can be synthesized. mRNA is same, and to that the so many ribosomes are there. Such structures are referred as the polyribosome, polysomes. Okay, means just like a beads seeds, one mRNA is there and it is reading, means one ribosome assembly is synthesizing one protein, another ribosome is, and simultaneously they are following to each other on the same mRNA. Depending on the requirement, what is the concentration of that protein should be needed, that accordingly the protein synthesis continues, and whenever a signal comes, it going to stop. Okay, so that's all regarding to these things. And what happens to the this particular peptide which is synthesized, because just here what we are saying that the one amino acid is connected to another amino acid is connected to another, in that way thousands of amino acids are get connected. But what happens, whether that structure becomes remains a linear one, or whether that structure undergoes a folding or all these things, that we are going to discuss in the coming lecture. So that's all regarding to the translation. In coming lecture, in tomorrow's lecture, we are going to discuss the post translational modification. Means how the once the particular polymer or the amino acid polymer or protein linear molecule is synthesized, what are the consequences happens, how it goes to particular location, whether it should be remain within a cell or whether it is secreted outside the cell, within a cell also where it is required. In that way, whatever what are the fates and how it is managed by the cell, that are the topics of tomorrow's discussion. So that's all regarding to the translation. Still a lot of details I have purposefully not mentioned whenever we are second time coming, because this is the basic knowledge sufficient as far as our syllabus is concerned, but for the advanced level examination we should go much more details, that also we are going to cover in. So that's all. I think time is also not there, hence bye bye for today. If any problem is there, I already given that we are going to conduct the doubt clearing session whenever whatever questions which you have, that you can explain me. Okay. Okay, thank you.